You will have 3 different wines to taste, and they are the same wine, the same base wine, which is fermented with 3 different strains. Okay? So, let's talk about the most simple type of strain that you might use to enhance your aroma in wine. And the first kind is the kind which is represented by Fermol Chardonnay. Fermol Chardonnay is the oldest of the strains I'm going to talk today. And this was developed under a very simple principle, which was to find a yeast which was able to produce fermentative aromas, so secondary aromas, not primary. And the way to do that, it's quite simple, which is passing through the Ehrlich pathway. So the Ehrlich pathway is a pathway that goes from the amino acids and basically transforms basic amino acids to higher alcohols, and these higher alcohols become esters. And so acetate esters and ethyl esters. And this is what does what many different strains of yeast do. And what they do is giving aromas which are based mainly on fruit, fresh fruit, banana, peach, and something like this. And this was the idea of Fermol Chardonnay, because we wanted a strain which was really Chardonnay-like, so able to give that kind of aroma which was mild, which was fruity, and it was really inviting. It was really captivating under the point of view of the tasting. Okay, and this is quite basic. And this is the first way by which you can face the enhancement of aroma in wine. It's quite simple, okay? You just have to find the right strain to do this, and Fermol Chardonnay is one of these. In the later years, we also have addressed another point of view, which was trying to improve the aroma of wines, not just concerning the aromas which are connected to the fermentation, but also concerning the aromas which are more related to the variety. So, in this case, varietal aromas. And if you think about varietal aromas, the 2 main classes of chemical compounds you can think about are terpenes and thiols. These are the 2 main categories you can think about, okay? Our idea here was to find yeast which was able to release a great amount of thiols, because we know that thiols are released, they are not produced usually, okay? It is not that yeasts are able to produce thiols, it is that yeasts are able to release the thiols which are already inside the variety, so inside the skin of the many different varieties. Sauvignon, for instance, has many thiols, a great amount of thiols, because the plant synthesizes thiols, not because the yeast synthesizes thiols. Okay? And our idea here was to do a breeding strategy, and the breeding strategy is basically you take 2 different strains of yeasts. Maybe one is interesting because it is really good at fermenting, and the other one is really good because it is good at releasing aromas, okay? But the problem is that it is really good at producing aromas, releasing aromas, but it has a really low fermentation rate, so it's really a bad yeast to use. What you obtain is a hybrid strain that has both the features of the 2 parent strains. So, in this case, we wanted something which was good at fermenting, such as PB2033, but we also wanted that new strain to have a good ability to release thiols and aromas more in general. And so what we did was to obtain many different hybrids, because the big problem with this kind of crossing, you cross different strains, but you get to this kind of grouping, so these 28 hybrids, and you have to search inside this group of 28 hybrids, which is the one with the good release of aromas. And our idea here was quite simple, and this is just like the way the whole production of thiols works. So what happens here is, this is quite important. You have thiolic precursors inside the skin of your variety. During the pressing of grapes, they are released into must, and during fermentation, yeasts are able to cut the thiols from their conjugated part, which is usually either cysteine or glutathione. And in this way, they are able to produce these aromas. So you have 4-MMP, which is more similar to box tree or tomato leaf. The aroma is more or less that. Or you have 3-MH, which is the one which is more tropical. Or you can have 3-MHA, which is something in between. No, sorry, this is more citrus-like, and this is more tropical or rose-like, flowery. Okay, this is the way these yeasts work, and we wanted to find yeast that does this. A yeast which is able to release this kind of thiols. At this point, you can do many different things. I think that the best thing would be just to find out by measuring with a machine which is the yeast which is able to produce the greater amount of thiols. But when you have 28 different strains, the best thing that you can do, since we are all wine drinkers, wine lovers, but especially wine technicians, is to taste the wine that you are producing with these yeasts. And so what we were able to do was to find out that, to find the strains that inside this group were the best at producing some perceived aromas. So not measured, but the ones that you are able to perceive with your nose. So it was a tasting arranged such as this. There were many different people tasting wine and just recording on a piece of paper what they were feeling, what they were smelling, what they were tasting. And so they were able to find a subset of strains inside these 28 strains. So out of 28 strains, we got to a small number of strains. These are like 6 strains which were the most aromatic. And it was not based on the machine. This was an experimentation we did with an institution which is called IFV, Institut Français de la Vigne et du Vin. Here you can see all the descriptors. So for instance, these strains were selected because you can see peach, you can see fine nose, so like the way the fine smell, peach, banana, all descriptors which were good and were in the direction of thiols. Okay. Once we did this, okay, we said we had these 28 strains. We discovered that inside these 28 strains we had 6 strains which were really good under the aromatic point of view, but we also want to select only those strains which are also very good at fermenting. So we wanted to avoid completely to bring to the market all those strains which had a low fermentative ability. Okay, what is this? This is a robustness test. What this does is taking the strains that you have selected and to try them in a must with a really low amount of nitrogen. And so by trying this kind of trial, what you do is find out if you have some strains which are weak and that you have to exclude. I don't think you would be really happy to use a strain such as this, which has such a low fermentation rate, such a low fermentative aptitude. And this is why we did this, so that we were able to exclude these 2 strains and we were able to select these strains. At this point they were 4 strains, but we ran many different trials and we saw that there were actually just 3 strains, only 3. And you are seeing only 2 of them that were both strong at fermenting and good at producing aromas. So at this point we have 2 strains tested only with human nose, and at this point we can actually take this strain, run another fermentation trial, and after this fermentation trial actually see how much of 3-MH and 3-MHA they have produced. So in this case we used a machine to detect it, and what we saw was quite interesting, because as you can see from here and here, these 2 hybrids, which are called hybrid 101 and hybrid 121, are better at producing, at releasing these 2 kind of thiols, better than one of one of our other yeasts which are quite good at producing thiols, which is Levoulia Hesperide, but also they are far better than this C strain, which is one of our competitors, a very famous competitor, which is quite good at producing thiols. Okay, so this was a great result for us. Okay, but this is not the only thing. Okay, the really interesting thing here was the fact that we used this machine only at the end. Before we used only the human nose, and you might think that this is quite at the beginning I thought that this was wrong, okay, because the human nose is not able to give you any numbers concerning how much aroma is produced by each yeast. But I think this was the actual force of this kind of strategy, because it's not machines that will drink wine in the end, it is the tasters. And this kind of approach gave us some advantages, because the human noses were able to select not only two different strains which were different under the thiolic point of view, but here you can see something that we have done in our lab under the supervision of Carlo, which is measuring all these compounds. So acetate esters, ethyl esters, terpenes, higher alcohols, they were all measured using our GCMS, so our lab equipment, and what we were able to see was the fact that we had selected strains which were not only different concerning thiols, but all other categories of chemical compounds, of sensory active chemical compounds. By using the equipment in our lab, so our GCMS, we saw that our selection was also able to select different strains which were different not only for the production of thiols, but also for many different aroma-active compounds such as ethyl esters, acetate esters, higher alcohols, and terpenes. This is quite difficult to see, all of these histograms, all of these columns. So what I did was taking this kind of descriptor and try to do a grouping by using a spider graph, which is far easier to see. You can see how different they are one from the other. One is the first strain I told you about was hybrid 101, which is now commercially called Fermol Lime, and the other is strain hybrid 121, which is now called commercially Fermol Tropical. And you can see how different they are. So this gives us once more confirmation of the fact that it is really important to have an expert tasting panel to validate this kind of yeasts. Right now I'm over with the presentation, and I would just like you to focus on this kind of graph. Here, me and Carlo talked together. We took the data from many of our tastings based on these different strains, and these are the different ways by which these strains are able to express the character of wines. Okay, so if you see here, Fermol Chardonnay is more based, as I was telling you, on the production of esters, and so esters give you some particular descriptors, peculiar descriptors, which are more towards the flowers and towards the fresh fruit, because it's a more classic strain. It's quite classic for Chardonnay, for aromatic white wines. The other two are quite a bit different, because they are able to give you some descriptors, some sensations that lean towards tropical fruits, especially for Fermol Tropical, tropical fruits, Fermol Tropical, and the other one, Fermol Lime, towards citrus, so grapefruit, lime, lemon, orange, and so on. Let's say that both of them give you a little bit of box tree, so the tomato leaf trait, but I have to say that the tomato leaf trait is more related to the actual variety you are fermenting. I mean, you need an especially thiolic Sauvignon to express a great amount of box tree character. Usually it's quite difficult to have it, let's say, in a Chardonnay or in a Trebbiano or whatever. By the way, this is Trebbiano juice, okay? This is Trebbiano wine, okay? So you will not smell the box 3 descriptor here, okay? But I don't want to force the. It's always wrong to tell you what you will smell in this tasting, but from now and then, as you are tasting and as you are thinking about what you're tasting, just give a look to this chart, because I think that it is quite indicative of what these strains do during the fermentation.